Hey, morning everyone. Welcome.
Yeah, hi everybody. Welcome back to
Yeah,
workshop.
today is our sixth sixth workshop in our Friday hands-on workshop series, and we are talking about plant phenotyping and tools and new technology about, for example, data science and plant phenotyping again. And it's been so fun so far. So if you are new here, go here and subscribe in our channel. Every Friday we are having new video and a new workshop. And also give your like because it helps us to send this information for more people and help us to help more people. And that's the main idea. I'm Felipe Matias, and this wonderful lady here
I'm
is
Annalisa Marrano.
Anahita Marrano. And we are the PhenomForce. Thank you
Yes.
for being here today.
Yeah, so as every Friday, I'm gonna start with presenting who we are. So as Felipe said, we are PhenomeForce. We are a community of people. So if you are already following us, you are part of this community yourself. And so this community aims to kind of put in contact beginners and experts in plant phenomics and like providing information to start your own project. If you don't know where to start from or how to analyze your data, we want to provide people the knowledge that they can use to do that. And so we are kind of new, but we have already a lot of followers. And so if you want to know more about us, you can follow us on our social media. For example, we have a Twitter account at Force Phenom, so you can follow us in general there. We share our information. We also have a website at GitHub, and we also have a Slack workplace, which is so for people that are not very confident with Slack, it's an app that allows people to be in contact and to share information. And so if you want to be part of this workplace, you just send an email to phenomforce@gmail.com. So in the last week, we had more people interacting, and we are happy of that. But we want to have more people like posting information or introducing themselves. Because as we say, like we are a community and we want to know you, and especially like we want people to interact with each other and collaborate and share information. So here you have the typical window of Slack, and I'm highlighting 4 channels that we think can be useful. First of all, there is the Friday and ends-on channel where you can give feedback about this workshop series. And also, like, there is the suggestion here below channel where you can also propose topics for the next events, because this workshop series actually is lasting till the end, more before till Thanksgiving, but we are already thinking about organizing seminars or other events for the next year. Also, like there is a channel called Introduction. So when you will be invited to join this Slack, please introduce yourself, tell the people who you are, what you do. And then there is also, we introduced this channel called Job Offers, because as me and Felipe, we are postdoc, but I think many people probably they are looking for jobs, both in academia and industry, and I think we can give support also for that. So if you are looking for job or you are looking for people, you can post your advertisement there. And so then we are at the, as Felipe say, we are at the 6th seminar workshop in this workshop series. We had already have like 5 amazing workshops. The major goal of this workshop series is to introduce people to open source resources for data analysis in phenomics, which we think is the most challenging part. We organized this workshop series thanks to the supports of the MPPM, in particular of Jennifer Clark and Christoph, and also thanks to the support of the Midwest Big Data Hub. And we also thanks Noah Fahlgren because he helped us with all the technical issues and the binder. And today is actually one of the speakers, so you will actually see him live. And yeah, and so I think Felipe is going to talk about who is gonna talk today.
Yes. So, guys, first of all, today we have many speakers, and we are using internet device and internet and new technology device, and all the time something can go wrong. So, if someone lost the connection or we lost the connection, please stay there. We're gonna do our best to come back and
Mhm,
continue the workshop. Okay. So for the people that register for this workshop, you receive the link for this
Thank
workshop. Yeah, exactly. And so please go there right now and press the button binder, as you can see here. Okay, this red arrow, and it's supposed to open up. Okay, Anna. It's supposed to open a Jupyter notebook from where our workshop is gonna be today, right? So, but then the speaker is gonna tell which one you guys should open. But and after open the Jupyter notebook, go there and press these two arrows, and supposed to open these two nice
you.
pipelines from where you're gonna run the code today. Okay,
Mhm,
and with this, I'd like to introduce our speakers. We are really happy to have them here today with us and being part of this project. Actually, PlantCV is quite actually super famous because many people contact us asking many questions and excited with this possibility. So, guys, it was one suggestions we brought for you. Continue giving us suggestions about nice workshops and nice people. And also, if you have a pipeline or software that is working well for you, for your group, for your team, and you want to share this with our community, please send a message to us. We can try to schedule
mhm,
a new
mhm,
workshop for you too. Okay. So the workshop today is an introduction to image analysis working for with PlantCV, and we have Malia, Noah, and Halle. All of them is from Donna Dunford Plant Science Center, and
mhm,
they're going to introduce better themselves now. And I
mhm.
would like to invite Malia, which is going to be our first speaker today.
Yeah.
Hi,
Hi,
Malia.
Malia. Hi.
Hi, everybody.
Yeah, we also want to say that if you have any question, you can type them on the live YouTube, uh, and we will interrupt the speakers to, so if they can, to answer your question. So feel free to ask anything that you want, because this is a great opportunity. You are the three developers of PlantCV, and it's a good opportunity to ask any question that you have. Yeah, so I think, so should we put also Noah and Hailey just to introduce them very quickly?
Yeah, sure.
and and then I think Malia is gonna start with her presentation, general presentation about PlantCV, and then I think it's Noah and Hailey, right? Or the other way.
It'll be Haley and then Noah.
Okay, perfect. Okay, so I think you guys, if you can start, and we will leave Maya. And good luck,
Okay.
and see you later.
Okay.
Okay. So can
Awesome.
you see my slides?
Yes, we can. We are gonna put them. Yes. Oh,
Not yet? I'm at full screen, so okay, let me.
yes, we can see them now.
Yeah.
Okay. So first, thanks to Philip and to Anarita for organizing this really, really nice workshop series. So I'm Malia Gehan. I'm here with Noah and Haley, and we're all at the Danforth Center, like Philip mentioned. And today I'm going to talk about PlantCV. I'll give a short introduction before we get to the hands-on portion in Binder. But first, I wanted to mention that the PlantCV project is supported by a number of funding sources. The main one being USDA NIFA. We have a FACT grant that supports the development of PlantCV, and we're really thankful for that.
Malia, just could you put your presentation mode or share the or change the screen?
Oh,
Mm-hmm.
Yeah,
it
we are.
was for me. Sorry. I I was in presentation mode. So is it in presentation mode now?
No.
Maybe
think it's from with the screen.
Let me stop sharing and then reshare.
Okay.
Yeah.
Maybe if you're using two, screens, you should.
Yeah, I
other.
wasn't, but I had been sharing it for a while, so let's try again.
Okay.
And then I'm going to go to presenter view and let me know if you do not see that.
Okay. I guess it's loading.
Is that working? Because I'm in. Okay, let me try. I was in.
Well, otherwise you can also continue like this. There is no problem.
Does this work?
Works.
Yeah.
Yeah, perfectly.
Okay. Alone on the screen.
Okay. So, like I mentioned, we're all at the Danforth Center, where we started the PlantCV project. So today, the goals for this session are to just give a really general introduction of the PlantCV project, if you've never heard of it, and then give some use cases for PlantCV. I'll, in one slide, briefly introduce today's data set, and then we'll flip over to Haley, where we'll actually do a hands-on development of a workflow. That workflow will include segmentation of the plant from the background, and then some new analysis measurements that have been included in PlantCV recently. We'll then show how to convert that Jupyter notebook into a Python script, parallelize it over a data set, and then actually analyze the results. So we're going from sort of start to finish, hopefully, in this workshop. But if we don't get there, then all the information will still be there, hopefully, for you to follow along. So at the Danforth Center, we're really lucky because we have a lot of resources for plant phenotyping. In 2013, they opened the Bellwether Phenotyping Facility. Can you see my mouse? I'm not sure.
No, I'm not seeing your mouse.
That's okay. So the Bellwether Phenotyping Facility is can house 1,140 plants. It can do automatic watering and weighing of the plants, and you can image the plants daily. This is a really nice system. It's very expensive, and so in addition to using this very expensive system, a lot of us at the Danforth Center also do low-cost imaging with Raspberry Pi computers, which are low-cost computers and cameras. And then we're also have a lot of other imaging systems at the Danforth Center. So my group in particular is interested in hyperspectral imaging, which is this system that's shown in the middle here. So because we're a research institute, that means that we don't, all the groups don't have a unified set of goals or research questions. We're also working on very different plant species with all different architecture types. So this is actually quite out of date. It's ended at the beginning of 2020, I believe. We've pretty much doubled the number of plant species on here because of Alison Miller's lab has put a lot of perennial species on here. And we've also accumulated a lot of image data. So millions of images just on the Bellwether system, and then many, many more on the lower cost systems that are sort of not included in this metric. So one sort of unifying thing for all of this image data that we're collecting, regardless of it being from a thermal camera or a hyperspectral camera or just a RGB camera, is that you're going to start off with an image. You're going to want to sort of pick out the portions of that image that are most important to you. So in our case, it would be a plant. And then you're going to want to extract information from that plant, that target object. So when we first started the PlantCV project, there were sort of limited options in what we had access to, especially what was open source. And what was available to us was a little more closed box than we wanted. So we wanted a way of being able to define traits, new traits, or simply understand what traits were being defined. So I like to give this example. So we had access to some software that defined plant height as this orange bar you see in the third picture here, where you have plant material at the top of that and plant material at the bottom of that, but a plant biologist would not call that orange bar plant height. And so for that reason, and sort of many other reasons, we knew that we needed a way, sort of a clear way to be able to add and define how the traits were being sort of characterized. And so the mission of PlantCV is really to create a set of phenotyping tools that'll be modular, reusable, so that they can be combined and recombined with ease to build flexible workflows to quickly extract biologically relevant data from images and sensors. And very importantly, wanted these tools to be usable by both bioinformaticians, data scientists, computer scientists, and biologists. So we knew that a lot of labs were interested in phenotyping, but might not have access to, say, a computer scientist working in their group specifically. And so these tools needed to be accessible to a wide range of people. One thing we noticed that when we sort of first entered this space was that we saw that there were a lot of tools available, sort of like ad hoc tools, but a lot of them weren't sustainable, meaning that, say, a graduate student or a postdoc would develop a tool in the lab, and then they would go on to their next phase in their career, and those tools would stop becoming maintained, and so then become less and less usable. And so with PlantCV, we really hoped that we spend a lot of time in developing infrastructure with the hopes that the tools that are built are reusable and reproducible. And that's important because we don't want the community, we hope that the community will be able to spend their effort in adding functionality to a tool base rather than having to reinvent the wheel every time. So one major goal of PlantCV is that it be community driven in its development so that we create mechanisms for the community to contribute to PlantCV. We want to make sure that the software can enable low-cost phenotyping platforms, because we know that a lot of researchers and research groups aren't going to have access to large, expensive phenotyping tools. And we wanted to make sure that PlantCV was documented well enough that it could be used for training and outreach for students and the user community. And of course, this workshop is a big part of that, and we're, again, really thankful for it. So PlantCV at its heart is a library of modular functions that you assemble into workflows. You then parallelize those workflows over sets of image data that can be tens of images, hundreds, thousands, hundreds of thousands, even larger sets than that. And then what you get out is a set of measurements extracted from your image data. Again, we don't want to reinvent the wheel, so wherever we can, we try and integrate with other open source projects. And so here are sort of hex badges of a lot of the projects you see. So the homepage from PlantCV is listed there. And so we'll likely be referring to the documentation, which you can access through that website sort of throughout this workshop. So like I mentioned, we hope to be very community driven, and a part of that is stable development. And being able to actually accept contributions from the community. So PlantCV is located on GitHub, and we spend a lot of time and effort, and a lot of our contributors spend a lot of time and effort making sure that the code that we add to PlantCV is documented. We have interactive documentation for all of the functions on PlantCV. And there's several tutorials for different applications. And then very importantly, all the code on PlantCV, or the very mass majority of the code on PlantCV, is tested, meaning that if I make a change to PlantCV that then breaks something that Noah has made, I can't just automatically push those changes and break PlantCV. PlantCV is available for download, of course, through GitHub, but also through PyPy, CondaForge, and there's a Docker version. We release versions of PlantCV frequently, so that if you're, say, publishing a paper, you know which version of PlantCV to cite, and there's sort of a stable version to kind of go back to. So PlantCV can analyze several different data types currently: RGB, grayscale, near, fluorescence images, thermal images, and hyperspectral images most recently. There's a lot of classical computer vision methods. We also have some graph-based segmentation methods that we're actually going to talk about today. That's one of the modules we're going to highlight that Haley has developed. There's methods for data visualization, machine learning, which I'll talk about a bit more. And we currently extract a lot of different measurement types. There are ways of adding custom measurement type extractions. And of course, we're always looking for more contributors to PlantCV. And so we welcome anybody who's interested to join our community. So this slide is like very egregiously out of date, but the community has grown since the first time we released PlantCV. So the first version of PlantCV had 3 developers, and the second version of PlantCV had this many developers, and we have many more now. So contributors to the development of PlantCV, and we're really thankful for that. So we often get asked how many people use PlantCV. That's kind of a hard metric to quantify. We know that we have 2,500 downloads of PyPy per month. We have 25 contributors. And really importantly to us, there's been 4 papers on the development of PlantCV. So if you're interested in contributing to PlantCV, you are included on that list of authors, which is always important for graduate students and postdocs. We're really excited that we have 19 user papers. So those are people that are using PlantCV in their research and have published on published. So here, this video is showing one of the interactive tutorials we have on PlantCV. For PlantCV through the documentation. We also have static documentation in case you find that more convenient. So there's 4 major steps of image analysis with PlantCV. You develop an imaging workflow in Jupyter, which is great because you get visual feedback on each of the steps that you're developing. In general, those workflows are broken up into 2 major steps: image segmentation, so the identification of the target object. And then once the target object is identified, you then apply analysis algorithms over that target object. And so we'll be doing that for the majority of our time today. And then we're going to demonstrate how you can translate that to a Python script, parallelize it over a set of data, and then do the statistical analysis downstream. So you might have had this in previous workshops, but it's important to go over, and that is just the basics of what an image is and what makes sort of image processing possible. So at its core, an image is an array. It can be a one-dimensional array or an array of X dimensions. So most of us, and the data we'll be analyzing today, will be RGB data. So that means it has 3 channels: a red channel, a green channel, a blue channel. And so you can think of that as an array with 3 stacked arrays. So a channel for red, a channel for green, and a channel for blue. So this is important to understand because when you're thinking about thresholding and image segmentation, it helps to understand a little bit about images and color spaces. So when you have an RGB image, say you have this image of this sort of dilapidated barn, if you have an image that looks like this to your eye, the top right image, you can actually separate that image into its 3 channels, and you get different intensities of red, green, and blue, which are represented by values like 39, 139, and 96. And so when you think of these images as simply as numerical arrays, you can begin to sort of understand how we can start to do math on those arrays. So there's more than one way to segment an image, but one of the more simple and traditional ways of segmenting an image is through thresholding. So in this case, we would take an image that's in RGB color space and then convert it to a different color space, say HSV or LAB, which is listed here. Select one of those channels. In this case, we're probably selecting either the A channel or B channel. So A channel stands for green magenta, and B stands for blue yellow. And the goal of this selecting one of these channels is to find a channel that you can start to segment your target object out of. So we see in this second panel on the top right that the leaf, the plant in this image is light. And so because it's light, we can then apply a threshold and say any values above this threshold set to 1 or white, and anything below that set to black. And that's kind of the basics of how a threshold works, which we can go over in more detail. And then again, once you have that and clean it up, you can then measure measurements over that target object. So we're going to talk a lot about thresholding today through Haley's workflow. We wanted to just also point out that there's not one way to bake a cookie or something like that. So there's more than one way to segment an image. So if you have the forethought in your image collection when you're collecting a data set to collect a background image, you can do something called the background subtraction on your images to try and segment out sort of your target object that way. There are also other methods within PlantCV that are really useful, especially when, say, you have plants that are under stress, and so might not be perfectly green, for example. And so Arash and Noah, several years ago now, included a really nice module in PlantCV, a naive Bayes classification module. That's initial use was to segment plant from non-plant in an image. So you could have category 1 be the non-plant pixels, and category 2 be the plant pixels. And then we calculate a probability that that one pixel belongs into one class versus another. So this does require some training, but it's very easy to train, I'd say, in comparison to, say, other machine learning methods that maybe are more complicated. But I think it's a really powerful method that we found several uses for. So just as an example of that, instead of doing just plant, non-plant, we can start to categorize an image into its parts. And so we had a collaborator who was interested in categorizing their wheat biotic stress images into healthy tissue, chlorotic tissue, and, say, pustules. And they wanted to get, say, pustule size, percent area coverage of chlorosis, percent area coverage of healthy tissue. And this is a lot more information than they were previously getting because they were traditionally scoring this data from a scale of 1 through 5. So again, it's easy to train this naive Bayes classifier. We generally use right now just ImageJ. You create a data set that uses their pixel inspector. You click on points, get those values, and copy them into, say, an Excel or a text file that has the categories that you're interested in. So in this case, it'd be plant, healthy plant, pustule, chlorosis, and background. And so using the naive Bayes classifier, we calculate the probability of, again, one pixel being in each of those classes. And what you get out is data that sort of looks like this. And so we can have a lot more information here, and it's a really powerful tool to sort of classify data fairly quickly. And so this particular tool has been used by other groups. So this is Tara Ender's paper out of Nathan Springer's group, where she's categorized her maze plants into stress versus non-stress pixels, calculated other shape parameters with PlantCV, and then calculated this over a full data set over time and across several genotypes. Okay, so in my last 2 slides, and then I know everyone's anxious to get to the exciting bits of actually doing hands-on. I wanted to highlight this morphology subpackage that Haley has added to PlantCV. This was a set of measurements that was highly asked for by the community. So now you can do all the segmentation using PlantCV tools and then calculate features such as leaf angle, leaf and stem lengths, leaf curvature, leaf insertion angles, and number of leaves using the set of tools that have been added through the morphology subpackage. And that's what we'll be doing today. So today's data set, we're actually using public data from Becky Bart's group at the Danforth Center that they published in 2017. And we didn't have the morphology tool available in 2017. And so, this is the opportunity to sort of reanalyze the data for a new trait. So today's data set, we've sort of sub-selected because of time limitations and size limitations. We sub-selected that data set down to 40 images, but there are many, many thousands more, and just 2 genotypes over 3 days. And so they planted different genotypes of sorghum. And so the question we're going to ask today is sort of which genotype of sorghum would allow for higher density planting. So I think most of you have already followed Philippe's instructions about going to Binder, but here's the GitHub site in case you missed that or need to go back to that. So if you go to that GitHub site, there will be a very small button to launch Binder. You click on that, and then I will actually stop sharing so we can get to Haley.
Okay, thank you for the amazing presentation. You already given many ideas how implement these actually in my research. That's awesome. So I'm going to invite Haley here. Hi, Haley.
Hi there. Okay, so I've got the GitHub page pulled up on my screen, if I can share that with all you guys. Like Malia said, so if you've got the GitHub page already pulled up, the Launch Binder button is going to be right underneath our title here. And once you click that button, it might take a few minutes to launch, but you'll get a screen like this. And eventually it'll launch a page that looks exactly like this. So we've got all sorts of resources so that you can jump back in at any point or pick up where you left off. But for today, we're going to start on the file that says build workflow here. So if I've opened that up, it's a mostly blank notebook. We do have, for anyone that wants like a completed notebook and doesn't want to keep up with typing along, the sorghum morph workflow is going to be the one that has all the completed steps. But for today, we're going to get started here. So the first part that I always do when I start a new Jupyter notebook, which is usually where I develop workflows, would be with import statements. So first, we're going to import PlantCV. And we're also going to import matplotlib because we use that for a lot of our backend plotting. The next part of the workflow, just for while we're in Jupyter, we can set the plotting size. So I'm going to increase the size for today so that hopefully everybody can see really well. And this is just, again, using the matplotlib backend that we utilize within Jupyter notebooks. And I'm going to set my figure size to a little bit larger than usual, which is going to be 8 by 8, but not in quotations. The next part that's already pre-filled out is setting up our class named options. This is going to be helpful when we go to, instead of running it in Jupyter, we're going to eventually run it in parallel. And this piece of code is going to be helpful for that. So the first thing that we're going to define is our example image that we're going to be working on. All of the images out of our data set are in a folder called sorghum images. And then the one that I'm going to use for an example today, I've got the path here. So I'm going to give the path from my current working directory to the image that I'm going to work on. Next, we've got a parameter called debug. And within PlantCV, there's 3 different debugging modes. You can either have none, which will just run the analysis and not plot anything. Instead, you can have it set to plot, which will plot it to the screen, or optionally, you can also set it to print, which will print an image out to the file. The last few things that we're going to fill out here is the results. We're going to save out our data to a text file just called results.txt. And then our out directory, I'm going to just set to our current working directory. Finally, the last bit of setup that we're going to do is a little bit of kind of fancier stuff in case you need to see things better. You can increase different parameters like the text size and line thickness. Those are all parameters that we can update within PlantCV. And that way we should have a nice clear to see image when we start running code. So the first thing that we'll do in a workflow is reading in our image. And it takes a parameter called file name. In our case, we're going to just use our argument that we set up, and it should pull this original image out of the file. Okay, so now that we've got an RGB image read into the notebook, the first thing that we really have to do at any workflow is separate plant from the rest of the background. This can be kind of intimidating at first. There's a lot of different ways to go about it, like Malia said. So we've added a couple of different tools that can help people visualize and get started with their notebook. And a lot of those functions are within the PlantCV subpackage called visualize. So those functions are pcv.visualize, and I'm going to show one that's the color spaces function. It takes an RGB image. We'll set it to the image that we read in. And optionally, it's got a parameter to show the original image overlaid with each of the color spaces, and I'm going to ignore that for now. So what this visualization function is going to do is show different color spaces separately, and that way it'll give us a good idea of which color space might be good for segmenting out the plant. We can see, for example, in the hue color space, there's a whole lot of noise within the chamber, and this is probably not going to be a very good option for segmenting plant from background. However, in the blue magenta color space, which is the B color channel of LAB, the plant is kind of popping out from the background because it's lighter than the rest of the pixels. So this is just a tool to kind of let you know which color channel would be a good place to start. So another channel that seems pretty promising is the saturation channel. So I'm going to start by thresholding that. And the first step of segmenting out is going to be transforming the color space of our RGB image into the one that we think is going to be the most appropriate. So like I said, we are going to start with the saturation color channel and see a little bit larger here now. our plant is significantly lighter than a lot of the background, although we do still have other objects that are just as light as the plant. So as Malia mentioned before, we use a lot of thresholding techniques. So I'm going to use a binary threshold here, and anything below a certain parameter will get set to black, and anything above will get set to white. And that way we can start to separate our plant out. So the input parameter for my gray image, I'm going to pass in the image that is just the saturation channel. I'm going to set my threshold to something to start out with. And the max value parameter within this function is what any pixels above my threshold are going to get set to. I'm going to set those to 255 for white, and then anything below is going to get set to 0, which is black. Object type, in this case, like I said, the plant is quite a bit lighter than the background, so we'll write in light here, but optionally you can do a reverse threshold and do anything that's darker in here. So one thing that I'll show that can be really helpful in the step above, I'm going to change my plotting mode from notebook. Well, this is inline within the matplotlib backend. I can change the backend to notebook, and it'll turn it into an interactive plot. And this is nice when I'm testing out threshold values, because I can interact with my plot by zooming in. It's a little bit slow. But you can also see when I'm scrolling my mouse over, it's going to give me in the bottom right-hand corner the XY coordinates as well as the pixel value. So I can see here that my plant pixels, it might be a little bit too aggressive to set it to 100, because some of the pixel values are more like 80 and 70, depending along the leaf. So I'll turn off that plot. And then with that information, I'm going to update my threshold from something like 100, and I'm going to try 80 instead. Now I can see I've included a lot more of my plant than I had previously. One caveat of the interactive notebook mode is that I have to turn plots off when I'm done with them. And for the sake of this, I'm going to turn that back off for now by changing it to inline. So now that I'm happy with this threshold that I pulled out of the saturation channel, I'm actually going to include another channel just to be really robust. The measurements that we're calculating today, it's important not to miss any plant pixels, but it depends on what your research question is. Since we're interested in the shape of leaves, any points where leaves would disconnect from each other is really detrimental to the algorithm, since it's sensitive to that. But on the other hand, if you were doing a project where you're interested in calculating NIR intensities from images, it would probably be a good idea to be more restrictive in the pixels that you include, because in that case, it would be harmful to include background information. But like I said, for our purposes today, I really want to make sure that absolutely no plant pixels are being lost. So I'm going to utilize a second color channel to really make sure that nothing's being missed out here. As I said in the visualization step, it seemed like the blue-green color channel was another good one. So I'm going to pull out that channel next from the LAB color space. So similar to last time, RGB to gray, but this time it's going to be a function that grabs the LAB color space, and specifically we're interested in the blue-green channel. Again, plant is quite a bit lighter than the background, And we could scroll over and really experiment, or you can also do trial and error to find out what threshold value is going to be the most appropriate. We've tested this out ahead of time, so I know which values are good. And I'll just fill that out here. Again, max value, I still want to set to white, so it's going to be 255. And the object is lighter than the background again, in this case. Okay, so pretty similar to the last binary threshold that I have. And I can see I'm pretty happy with how well connected each of my leaves are, since I definitely don't want them to be separate objects once we've got everything sorted out. Okay, and finally, to make sure our bases are completely covered, we're going to include just one more color channel. Again, out of the LAB color space. But this time we're going to use the green magenta channel, which is A. Compared to our last step, our last binary threshold, in this case, our plant is actually darker than the background. So I copied my thresholding step, and I'm going to just update it here. So in this case, I'm going to be operating on our green magenta slice, which is stored in the variable called A. I know that my image is dark, or my plant is darker than the background, and values below 122 are generally where my plant pixels are. And I have to just make sure that I rename the variable that it gets saved into. So now I've got 3 different binary masks that all have plant kind of segmented out, but each of them still have a lot of background. So the next steps that we're going to do is, by combining different channels, we're going to start getting rid of some of that noise. So the first 2 channels I'm going to combine together are the blue magenta and the saturation channel. And we can do that with PlantCV functions, or you can also use, if you prefer, like OpenCV for stuff like this. It's all very similar. So the 2 binary images that I'm going to combine are my S threshold and my B threshold.
Ali,
Yeah.
there is a question. Do you want to answer now?
Yeah, go ahead.
So Travis is asking, is there an integrated method for doing the segmentation method mentioned earlier for when you have access to a background image?
Oh, that's a really good question. I think Malia showed a slide. We do have a few other functions such as background subtraction, and that would probably be a really good place to start if you've got a clean background image for all of your, for each of your plant images, especially if it's got the pot in it. It can be really nice to be able to remove that early on instead of the way that we're going to have to clean it up here. The other option there is image subtraction. So instead of just background subtraction, you can try subtracting just manually and seeing if that does some good segmentation. Generally, we found that using color spaces is pretty robust. So.
Yeah, I think also Malia commented on the, maybe she wants to say also on the stream. No?
I'm answering questions in the chat, but go ahead.
Okay. Okay, cool. Thank you, Ellie, for answering.
Thanks. Yeah, just interrupt me as questions come in. I'm happy to take any of those. Okay, so the way that I'm going to combine them in this case is a logical OR. I mentioned already that I want to be pretty aggressive with how many the plant pixels that I'm keeping. And so I want to keep anything that was plant pixel in the S threshold, or if it was considered plant pixel in my B threshold, I want to keep both of those. You could be more restrictive and do a logical and on 2 binary images instead, but in our case, this is going to be the most appropriate. So you can see it's already a little bit clearer than it was before, and I'm going to be doing the same thing and add in our A threshold channel as well. So my first binary image I'm going to include is the output from the last one. And that's generally the workflow. It's a lot of the output from the previous step is the input to the next step. And my second binary image is going to be my A channel threshold that I created earlier on. As you can see, my plant looks really good, but there's a ton of background. There's a lot of the pot still being picked up, and we're going to take care of that next. A lot of the cleaning will be able to get rid of the salt and pepper noise if we do a fill step. And this is somewhat similar. This is pretty similar to the threshold step, but instead of thresholding on pixel value, we're going to threshold on object size. So with the PlantCV fill function, it takes a binary image as its input, and I'll give it our binary mask that we made, and it takes a size parameter. This says any objects located in the image that are less than 15 pixels large will get removed. So that'll get rid of a lot of our kind of salt noise that is along the edges. As you can see here, the fine salt is all filled in now. And you can be a lot more aggressive with the size of the fill that you're doing, but we wanted to do just kind of a initial cleanup of the image before we start using regions of interest to further clean it. So the next function that I'm going to use to help clean is a dilation step. This takes grayscale or binary images, and as the name suggests, it dilates the objects that are found within the image. So any object that is white will get a padding added onto it. It dilates and makes all of the images a little bit large, or all of the contours a little bit wider. So compared to our last step, you can see everything's about 3 times as thick along the edges. All right, and now I'm going to finally do a more aggressive filling step because our plant is a really large image now, and I'm not afraid of accidentally filling pieces of leaf in. So I can do a much larger filling step where anything smaller than 250 pixels gets covered up. So this is starting to get cleaned up nicely, but right now it's just a binary image. We need to get it into a format that that recognizes it as separate objects within the image. So the way that we'll do that is with finding objects. The image that it inputs is just for the debug plot that gets made. So I'm going to feed in our RGB image in this case, and the mask that I'm going to have it find objects from is our most recent binary mask. So now in pink are highlighted all of the objects that have been identified. Still a lot of background, but we're going to filter that out using a custom ROI. There's a lot of different options that we've got as far as regions of interest. It can be really difficult to get some of the noise out of edges. And so more important than just being able to filter on the color of pixels, we also want to use the location of objects to help us filter out noise. So you can do this with a rectangle. There's a lot of different shapes, a circle ROI, but in our case, I really wanted to use a custom region of interest because I want to just include the plant and really avoid the pot. And the way that I'm going to tell it the shape of the region of interest I want to use is by providing a list of vertices. So the way that I really want to keep my plant but avoid all of the soil is by cutting out an area like this. So the soil is always going to be picked up in our case, but I really don't want to lose any leaves that get long and might hang down over the sides of the pots if I can help it. So I can use different vertices and use their XY coordinates to create a custom ROI, which is really helpful. So I'll show you, I'm going to copy and paste that over from the completed one. And these vertices that get drawn are input here. So I start with, this is X and Y values. I believe the first vertex is this one. But then just in order, I provide the kind of edges of the region of interest that I'm going to use for cropping down and cleaning up further. Okay, so the next step is filtering on this ROI. I want to keep everything that's inside of my box while getting rid of all of the noise that's around the edges and inside the pot of the plant. So the way that we do that is with ROI objects, and this is one of the longest commands that you'll have to write. I usually copy and paste it over from an older project. In this case, I'll take it out for you guys. It'll give us 4 different outputs, including a list of objects, a list of the object hierarchies, which lets us know if it's a like an object or a whole within that object. The kept mask is going to be the binary mask, and then it also will report the object area of the objects that got kept. Again, the image input is going to be for plotting. I'll give it the RGB image for this. Our ROI contour is this ROI custom from the previous step. Our ROI hierarchy is also going to come from our previous step.
Hi, Ali. There is another question. Sorry. Would an alternative to identifying ROI be to crop the image? Is that a good alternative or usually not as robust?
Cropping an image is definitely possible, and we've got functions to help you do that. There's also functions where you can auto crop down onto objects. I would say it's generally not as robust because there might be information from the edges of the image that you'd need for different parts of your workflow. For example, a lot of images will have a size marker or a color card passport, like a grid of color cards for color correction. And if you cropped initially in your workflow those pieces out, it would not be as helpful. Another benefit of the region of interest is we can keep things that either partially overlap with the ROI, or we can also do a hard cutoff. In the case of this kind of funky shape that I drew, we wouldn't be able to crop the image to such a strange shape. It would only be rectangular. But definitely, as mentioned before, there's a lot of different ways to get the same result. So if that feels more intuitive, then that's definitely a good option too.
Okay. Thank you, Ali.
Thanks.
You.
Okay. The objects within the filtering step are going to be from 2 steps above, where I found all the objects in my image. So my object contours are going to be from that step, as are the hierarchies that I'm going to use. And like I mentioned, there's different ways that you can treat the ROI filtering step. In our case, I made a really funky shape and I want to cut everything out that's not within there, usually we will do a partial overlap, and that way we can draw just a small region of interest on top of the plant and be ensured that anything overlapping with that region of interest gets kept, while objects that are not overlapping at all will get filtered out. Oops. Okay, so this has got multiple debugging outputs. It'll show me the objects that get kept within my region of interest, and then my new mask is this object that just has plant in it. So we're gonna do just a final couple of cleanup steps. In this example image, it's really clean. I don't see any kind of detached leaves, but if I did have any detached leaves, it would be detrimental to the algorithm, which will extract different shape characteristics. So I'm gonna add a fill step here to make sure that if I did have any disconnected leaves, they would get removed. The next part that is kind of quirky about finding morphological traits is that if there are any gaps within our plant, when I go to skeletonize it, those small gaps will kind of blow up and get exaggerated. The way that I can handle that is by closing in any of those holes with a closing step. So again, the input into this function is my output from the last one. And our image is pretty clean, so there's not a whole lot of a difference. But when you're running over a very large data set, you would want to check on multiple images to make sure. So closing is similar to dilation, that it'll kind of add padding onto edges, but in this case, it only adds padding onto inner edges. More aggressive is to completely fill any holes. This does like a flood fill. And if there was any gaps inside of my plant, they would get completely filled in regardless of their size with this step. Okay, and then since I did a little bit more cleanup, we're going to repeat and find all of the objects. So I can go from a binary mask into talking about things as contours and hierarchies instead. And that's going to be with another find object step. Sometimes segmentation can be much quicker. For example, if you used a naive Bayes classifier, it might be 1 or 2 steps of cleaning up instead of lots of different filling things in steps like I'm doing here. Okay, and then again, I found all of the objects that I want to keep. In this case, it's just the plant, which is exactly how I want. And then I can use an object composition step to combine each of the contours into a single object. So now I've got my object outlined in blue. All right. Now I've got my plant completely separated from everything else. It's cleaned up, and once we've got the object segmentation step completely done, then we can start collecting different measurements from that. So the first thing that we're going to look at is just kind of the general shape, which we can do with analyze object. This records a lot of different shape characteristics, including height, width, how solid an object is, what the perimeter of an object is. And you can see here the debugging image shows kind of different things that are getting collected. The image that gets returned is this debugging image. And if you're interested in looking at like the data that actually got extracted from this step, all of that's hidden in the background.
Sorry. There is a question. Well, there are two, but I will start with the first one. Normally, do you take images from different angles for each plant, or how is selected the correct orientation of the plant?
This is a good question. The example images that I'm showing here are off of our bellwether phenotyper, and it's not oriented consistently. They, since they're on the conveyor belt system, it kind of ends up just being however it's positioned. But it does, we usually collect images, one image like this, and then it'll rotate 90 degrees and take a second image. So in this case, it's not manually positioned. I'm doing an example on one of the best positioned ones. But for the actual project using this data set, we did a pre-processing step where I compared the side view and front-on images of each individual plant for each day and selected which one I thought should be the best pose. And I did that with shape characteristics.
Okay, thank you. Yeah, so the other question is for Malia, I think. So she's replying live on YouTube, so you guys can also read. Also, like I want to read just one of the answer of Malia, if people are like she about these things of the orientation of the plant. So Malia say that the bellwether system does allow for capture of multiple angles. With other imaging system, that is not possible. So it just depends on the trait. Yeah. Well, thank you, Ali. You can go ahead.
Okay. All right. So as I mentioned, I'm returning a debugging image here. But if I actually wanted to get at some of the measurements that are being stored, all of those are being stored kind of behind the scenes, but they are still accessible if you did want to look at something. I'll show that a little bit later on, but we'll kind of continue on and get into the morphology part of the workflow, which is how we're going to start extracting information about leaf angle. As I mentioned in the beginning, I'm going to set my different parameters to make it more readable for the workshop here. So I'm increasing a lot of our text parameters to make them thicker. And the first part of the morphology workflow is going to be turning our mask into a skeleton. This is a iterative thinning process so that only the structure of the plant remains, but none of the thickness of the leaves is there anymore. Everything's just 1 pixel wide. Okay, so it's probably very difficult to see. Just 1 pixel wide, I've got a structure of my plant. I can zoom in for you guys so it's a little bit easier to see. And this is my skeleton. Like I said, the lines are just 1 pixel wide, but it completely maintains the structure of the plant. And this is what we're going to use to start measuring traits. So one of the things that usually comes up is any kind of rough texture of the perimeter of the plant might get detected as important structure, and they don't want to lose that information. So sometimes you'll have barbs along your skeleton, like you would find on the stem of a thorned rose, and we're going to be able to remove those by pruning them off. So this step takes any segment that is smaller than a set size and removes them.
Hi, Haley. Sorry.
Hi.
I feel really bad every time interacting.
No need to apologize.
Yeah, so Leonardo is asking, does this workflow work with a loop through the images? But I think that's something that probably, yeah. Do you want to
That's a good question. Yeah, if there are any closed cycles in this step here, like I mentioned, if there was like one missing pixel in the middle of the plant, that loop would get exaggerated by this step, and you would see a big hole in the skeleton. And it's true that those loops are really detrimental for the next part, the algorithm that is going to start sorting segments into leaf versus stem. It doesn't really know how to handle closed loops like that. But since that is an important consideration, we do have a function that will detect any closed, it's called detect cycles, I believe. And that will look in your image for any of those closed loops that could cause problems down the line. But I would definitely recommend filling them in ahead of time if possible. The other reason that comes up is when our plants get big enough that leaves start to obscure each other. You can get closed circles that way, and that's another limitation.
Yeah, so also Malia say that PlantCV provides also has function for parallelizing a workflow across many images. So if you have your entire workflow, you can use it. You can use this function to apply the workflow on all the images that you have in a folder, for example. But
yeah, definitely. That'll be. I'll pass it off to Noah here soon, but he'll take it from developing the actual workflow to executing that over just a set of images in parallel.
Yeah, and yeah, there is another question, but I think Malia replied already somewhat. So, but I don't know if you want also to. I don't know if you. I think Malia replied already, but so it should be okay. Okay, thank you.
Okay.
Oh,
cool.
well, there is another one now. I will. Sorry. Does the skeleton eyes pick a pixel from the middle of the stem leaf, top, bottom? Is there some distortion issue here?
That's a good question. The function within PlantCV is based on a scikit image function. So we're utilizing their algorithm for skeletonization. And what the algorithm is doing is an iterative thinning. So it'll remove the perimeter over and over again from my plant mask. And the pixel that it keeps is just whatever one was in the middle. So it'll continue to remove structure, thickness on the structure until it's 1 pixel wide, and then it'll stop removing from that portion and continue thinning the rest of the structure until everything's just 1 pixel wide. There can be definite distortion, like I mentioned, if there's a really jagged edge to the perimeter of a plant, that might be recognized as important structure, and you would get kind of like the prunes sticking off from that. We've got an example on our documentation page. But a lot of those issues we can just handle within our workflow.
Okay, thank you, Ali.
Thanks. Okay, so like I said, pruning off any of those kind of extra bits is going to happen during this step. And it takes any segments that are smaller than our specified size, so let's say 30 pixels in length, and removes them. And this will plot out 2 different images, actually possibly 3. It'll show which objects got removed, which objects were kept, and then it'll also break our skeleton into different segments. So now I've got my plant broken up into different bits here. The next step, since I've got just the structure pulled out the way that I'd like it, our next step is going to be doing, I like to add in a second pruning step actually, because sometimes there are just unnecessary image data on the tips of, or if there are any branch points that got removed, it can be a good idea to just do another second pruning and make sure that there's no small segments that got left behind. From there, I'm going to sort all of these segments because I'm, for our workflow, I'm really just interested in leaf characteristics instead of also measuring things about the stem, because those are getting identified right now as well. So the way that I can sort that is with our segment sort function. And it'll return 2 different lists of objects, those that were identified as leaf and the ones that are, or I should say, primary and secondary segments. In our case, primary segments are going to be belonging to the stem, whereas secondary segments are going to belong to leaves. For my skeleton image, I'm going to provide it my pruned image. Okay, so after sorting, the debugging image is going to show me how the different segments got sorted. Anything that belongs to our primary group is going to be labeled in pink here. And I can see that it's doing a good job of separating out our stem from the leaves. And the way that this sorting is being done behind the scenes is by considering the branching points here. So if a segment has got a branching point and a tip point identified, that will be sorted into a leaf, versus if a segment has got branching points on both ends, that's going to be sorted into stem. So now that I've got that separated out, I can start to kind of look at information about just the leaf instead of measuring things from all identified segments. Okay, I'm going to go through this part a little bit quickly. I'm copying and pasting lines of code out of the completed Jupyter notebook, and you can feel free to do that as well. So the next step that I wanted to include was IDing the different segments. The way that things are numbered are in the algorithm are generally kind of bottom to top. But it would be important to know which ID matches with which leaf, because you might only be interested in a subset of the leaves. But this image will let you know the order in which segments are getting measured. The measurement that's of most interest for today is the relative leaf angle. And so the way that we can do this is with the segment insertion angle function. It's going to accept a skeletonized image, which we're just providing our pruned skeleton as before. It's also going to ask for a image, and this is going to get used for plotting. I'm going to use the output segmented image from my last step and put that into the segmented image parameter. Additionally, it's going to ask for leaf objects and stem objects. I'm going to provide those 2 lists that were output of my sorting step. And then this last parameter, size, is going to be informative in, it likely will change mainly depending on the size of your image. The way that these measurements are recorded is it will fit linear regression lines to just portions of each segment. So the way that we measure the stem angle is by fitting a linear regression line just to the bit of the leaf that's really close to the stem. So I'll zoom in here so I can show what I'm talking about. In the example of this really light blue leaf, I can see there's all of this is identified as one leaf segment, but I'm going to use size 22 pixels from the inside corner here to draw my regression line. And this green line is going to measure the angle between this thicker gray line, which is fit to the stem. That way we can look at the angle between the stem and the leaf that's emerging there. So it's also plotting the degrees that got identified for each leaf. And as I mentioned before, all of the stuff that's getting collected is being stored in the background as well. So that's the kind of the trait of interest that we're going to do some data analysis with today. But there's a lot of other functions that will collect information about the plant as well. One of our team members recently added a function, and it fills in the segments. So I passed it just leaf objects here, and you can see it does like a flood fill of each of the segments, and this is going to allow us to measure things like leaf area. Something else that might be interesting to researchers, just depending on what traits are of interest, you could collect the length of each of your leaves. This is measuring the path length. So it measures the entire length of each leaf segment. You can also measure things like the Euclidean length, which is going to be tip to tip. And instead of measuring the entire path, it's measuring, it's 317 pixels between the kind of insertion segment part of my leaf and my leaf tip here. So that might be of something of interest, depending on what the question is. These different used to calculate kind of a curvature of our leaves. So if my path length is significantly longer than my Euclidean length, that will give me a value larger than 1 when I take the ratio of them, versus leaves that are very, very straight are going to have values that are very close to 1. So that's another thing that you might be interested in. Instead of doing the relative leaf angle, we can also do a just straight segment angle. Again, I'm passing just leaf objects because that's what I'm interested in today. And compared to before, where I had a size parameter where it's fitting a regression line to just a portion of the segment, in this case, it's fitting a regression line to the entire segment. So you can see here, this green line got fit to all of the points along this curve. So just another way to measure leaf angle. We can also measure the curvature of a leaf in a different method by kind of utilizing the regression lines again. We can fit lines to both ends of a leaf and look at the angle of curvature. So as regression lines are drawn to each end of the leaf, it's measuring the angle between those two lines. So if something is very close to 180, it's going to be a very erect leaf, versus if you've got an acute angle, that would indicate a lot of droopiness in your plant leaf. And this type of stuff usually takes a little while to run because it is computationally expensive. Okay, like I said, it's fitting a line on both ends so that we can look at the curvature angle. There. A newer function that we added is the ability to analyze different characteristics about the stem. So in this case, I'm going to pass in my other objects instead of my leaf objects. And this is going to just collect information. Instead of about individual stem segments, it combines them into a single object and recognizes that we can measure the height and angle and different characteristics that are specific to stems. Finally, we can print all of the data that got collected out to a text file with pcv.printResults, and the file name that it got saved out to is going to be, in our case, I'm going to pass it whatever we saved our results file to. So if I go back one step here to the landing page of the Jupyter Notebooks, I can see a new file called results.txt, and it's got a whole lot of stuff that was all collected during this workflow. So if there's any questions on building a workflow, I can answer those now. Otherwise, I'll pass it on over to Noah, who's going to take this workflow from a single image and run it in parallel.
Yeah, thank you. Thank you a lot for your presentation. It was really, really great. Thank you.
Thanks.
There is a question, Ellie. Wait. So she's asking, do these leaf morphology aspects work with compound leaves as well? For example, tomato?
So far, the species that we've been using these tools on have been mostly grasses. We did a validation kind of experiment with a bunch of rice plants, and then I'm working with sorghum here. I also have done this with maize. All of those work great, but the types of analysis that you might do on like a tomato plant maybe wouldn't translate as well, but there's everything's so flexible within PlantCV. Depending on what the characteristic is that you're interested in, you could probably utilize these same tools and algorithms for your own purposes.
Another question is the measurements, the unit of the measurement. It's in pixels, but for example, you can have in centimeters or millimeters to compare with, you know, different crops or experiments.
Yeah, that's a really good point. I don't think we mentioned there is a function report size marker within PlantCV. So that's really kind of helpful if you've got a size marker in your image that you know the area of. You can translate from pixels to like centimeters squared or something like that. Some of the measurements that I showed just now within the morphology subpackage are going to be in degrees for the different angle measurements. But that's true. Most of the things are in pixels. So we can translate from pixels to area, or if in the case of the bellwether phenotyper, I've got a consistent zoom level, I'm able to compare measurements that are all pixels with each other.
For example, for my seed project that I didn't have like any differences in the zoom or like height of the camera, I think there are also like kind of a coefficient that you can convert from pixel to centimeters based on the resolution of the image. Like if it's 300 dpi, then you can convert somehow if you want to convert that and compare with other paper.
Oh,
So,
that's really cool. Yeah, I would usually recommend a size marker instead of a straight conversion, but for sure there's many ways to do it. And if you've got a color card in your image, we can also collect like an average chip size, and that way you can normalize if your images are not the same zoom level.
Yeah, that's a great suggestion. Another question is, it is possible to adapt this pipeline for root systems?
Definitely. Like I mentioned, whenever things start to obscure each other and have closed cycles, and that happens early on in roots, that's problematic when it comes to sorting out the segments from primary root to secondary root. However, we've done a little bit of work with hypocotyls, and it's pretty exciting to see that we can segment out like primary versus secondary roots on those images. But yeah, once the plants get a little bit more mature, things break down pretty quick. So
okay, so great. So let's continue with the workshop.
Thank you,
I'm going to invite Noah right now. Hi, Noah.
Hi. Hi, Noah.
Can you hear us? Yeah. Your volume maybe it's pretty maybe low. Could you talk?
Yeah. It's a bit low.
Great. Yeah, and one question that we always receive is the possibility to adapt, for different crops and roots and.
Yeah, actually, I'm a PlantCV user, so I use it for seeds in rice. But now we are also using for cranberry images, like in the field. So I think the cool advantage is that it's very flexible. Of course, you have to adapt the workflow on your plan. So I think these workflow are really useful because they help you to understand what the function they actually do.
Exactly. Yeah.
But then you have to adapt. Well, can you talk? Yeah.
Is this better? Okay.
Yeah, better. So we are gonna share your screen and then we are gonna leave you.
Oh, yeah, no worries.
Maybe you should increase a little bit the size.
Yeah. I'm not even going to use this window, or not this page, but yeah. One thing I just want to remind everybody that in the binder environment in the GitHub repo, the workflow Haley built, she built from scratch with this, the notebook at the top, but the file morph workflow notebook has the completed version. So we know we went through it quick, but if you want to look back at the completed version, it's there. Also, some great questions about using some of these tools for other things, and we just encourage people to try or reach out and send us some example data, and we're happy to take a look. You can sort of use any of these kind of like image analysis tools on just about anything, so it's worth experimenting a little bit. And oftentimes with new data, we realize we need new tools, so we're always happy to think about that. All right, so Haley showed how to build the workflow in a Jupyter notebook, which is great for visualization. And the question came up about, you have a whole data set, and we've walked through manually like one image here, but how do you then apply this to a whole data set? So we have some tools in PlantCV for batch processing, and the way it works right now is we need to be able to run this workflow as a Python program or a Python script. So what we do then is convert the notebook to a script. And so typically you might just do that by, if you click on the file menu, you can select the download as submenu and then Python. And that will download, it's going to open a blank page here, but it'll download a Python script file, a version of the notebook to your computer. Because we are working in this cloud environment, though, it's going to download it to your computer, but that doesn't get it for us in the environment. So what I'm going to do is click on the new menu and then terminal. And then I can run the command Jupyter notebook. And actually, I don't remember what the command is actually. Oh, wait. That's Jupyter NB convert, I think.
Noah, excuse me.
Yeah.
It is possible to increase a little bit the size?
Yeah.
Okay. Oh, yeah, that's perfect. Thank you.
Right. So sorry, let me clear that just so it's cleaner. So it's Jupyter NB convert name of the notebook, and then I'm just going to say test.py right now because we already have the completed workflow Python script available. Did I do that right? Sorry, actually, I don't know if I did. Oh, anyway, sorry. Don't need to mess with that too much. I'm going to open the Python script. The point is you'll get a text file. It's a Python file, but it's just a text file, and it will have all the contents of the Jupyter notebook just in text. So it'll remove all the outputs, and you'll just have the code parts, and there will be the anything you commented or the workflow we made available. And the GitHub repo has a lot of like formatted text for our explanation of what each step is about. That'll all be in there as comments, which is these lines that start with hashtags. So those are just ignored by Python. And I'm not going to go through this extensively, but in order to convert the workflow right now from Jupyter to a script that we can run, there's a few esoteric things we kind of need to do right now. So we're going to import a package called argparse. This is going to let us collect inputs from the command line. And in the notebook, in the Jupyter notebook, we had a section where we had a little Python class called options. We're just replacing that here with a function called options that uses argparse to get the inputs from the command line instead of hard-coded into a notebook. And this section of code is on our documentation page in the Jupyter tutorial that's on the sidebar of the documentation page. Yeah, so I'm not going to go through the details here, but the same options were hard-coded into Haley's notebook, and so we're just getting them from a different place. The rest of the notebook, we're just going to create another function called main, and then we just highlight all the remaining code in the notebook. And in the Jupyter text editor here, even I can do this. If I hit tab, it will indent. So this is already indented code, but this would all start like over at the left, and I'm just gonna use tab to indent it under the main function. But I can highlight the whole code block and indent it all out in one command. So that's already done here. And I will mention in putting this together, we did discover a few little bugs we need to work out in some of the ways we handle outputs. So I do have a little hack coded at the bottom. We wouldn't expect that you would normally have to do this, but just so you know that I added in a little something to make this work right, that we will fix in the future. So, PlantCV is never done. The last thing we need is just a little bit of Python code that goes at the bottom of a Python program like this. And again, you can just copy this from our documentation. But at the end of your script, you'll always have that. So then if you use the options thing that we describe in the Jupyter notebook, you won't have to change any of the rest of your workflow because you're using this sort of this args nomenclature for all of your variables. So args.image. If you hard-coded anything in your workflow, like file-wise, you would have to replace that to make it compatible with input variables. So I know this part's kind of weird and wonky. So, if you get stuck on this in the future, just let us know. we have some ideas about ways to improve this, but for now, it's just kind of how we handle it. Okay. So if we pretend that we went through the process of converting our notebook here to a Python script here, what we need to do batch processing is two things. And the first part's, this part's kind of new, but I'm going to open this notebook I put together. It's real short. And all it's about is we have a new system for using a configuration file to set your batch processing parameters. And so I'm just going to import PlantCV.parallel. It's a subpackage of PlantCV. And then we have this built-in method called workflow config. That's going to create a configuration data object. And then one of the things I can do immediately is save it to a file. So it saves the file in a format called JSON, which is just a hierarchical data format. And what I'm doing here is like, this is just an empty configuration object. And by saving it, I'm really just saving a template for what our configuration file would look like. So this is already in the GitHub repository. And so after I save the template, what I would do is go back over to here. And then if you click on it, so it's sorghum morphology workflow config.json. It pre-populates, a lot of these things would be empty, but it pre-populates what the configuration file is. So it's a bit long, and you can ignore all the bottom part because the metadata stuff is just hard-coded into PlantCV. You don't have to change that. But the stuff you do need to set is the input directory. So where are your images stored? In this repository, they're stored in a folder called sorghum images. So I've added that into the input directory. The JSON parameter is the name of the overall output file that we want to create. So that's sorghum results.json. The file name metadata, this describes the metadata terms that are used to compose the names of the images. So we process, when we go into the directory and we find image files, we will take the delimiter defined here. It's defaulted to an underscore, but you can change that. We'll split the file name up on that underscore and then break the image file name into the component metadata terms, and we store these. So these terms are described below, and they're just like a consistent vocabulary we currently use to describe like information about images. If you looked back at the sorghum images folder, they have these really long names. We don't need to go into what all of it means because they're kind of specific to our Lemnotext system, but this is how we're defining them. So image type, camera, et cetera, et cetera.
Hi, Noah. There is a question.
Yeah.
Ken is asking, could you elaborate a little bit on what sort of issue can be caused without the workaround? When is the fix necessary? And I think he's referring, I meant the code lines added to the .py file to make something work right.
Sure. So in just a little bit, I'll show you how we convert we get this one output file in JSON format, but that's not very convenient for doing statistics. So we convert it into 2 CSV files so that you can use it in R, for instance. Mm-hmm. So one of them is straightforward. We call it single value traits, for lack of a better term. It's when you have like a single measurement. So like a area of the plant is just one number. But then we have the second one is what we call multi-value traits. That's where you have the measurement isn't just one number. So like you can imagine, well, like when we analyze color, we get a frequency distribution of the hue values of the plant, for instance. So the morphology workflow that Haley described, a lot of those measurements, you get more than one measurement. So you get the angle of every leaf in the output. So for boring reasons, some of the things that we output multiple values of are coordinates, and those weren't designed to go into these CSV files, but our current code is trying to put them in. And that's like a bug. We need to like handle coordinates different from these multiple measurement outputs. So anyway, like if in the hack I put into the workflow, I just remove the outputs that have to do with coordinates. So like one of them is the coordinates of all the leaf tips, and another is the coordinate of all the branch points.
Okay. Yeah, thanks, Noah, for answering.
So in order to get like your metadata into your outputs, we have to be able to get the metadata from somewhere. So this is how we extract it from file names. And then we define the name of the Python script we created. This is what we call the workflows. So that's just the path to the script. And then a few other things like an output directory for image files. So this is if you save like any of the pictures that Haley was showing in the outputs, you could save into this directory. And a bunch of other things that I won't talk about now, but gives you some flexibility in terms of filtering on, like if you don't want to analyze all the images in the folder, you can filter on various criteria. The last part, it defines the parallel computing environment that we're going to use for batch processing. So here we're using what we call local cluster. That just means we're going to do the computing directly on the server that we're sitting on. And the only thing that really matters in that case is this n workers property. That's the number of parallel processes. So we're going to just use 2 because Binder's a limited computing environment. But in the documentation, we describe how we support, this method supports other clusters, so htcondor or Slurm, and several others that you can replace this with, and then configure it to run on your cluster with some minimal inputs here. So once you have the configuration file, so you would just edit that file, save it. Don't need this now. And we've got that file here. So then we're going to go to the command line. And the program that comes with PlantCV is called plantcv-workflow.py. And the only thing we need to give it is --config space sorghum morphology workflow config.json in this case. So just --config space and then the name of your configuration file. For time purposes, I'm not going to run this. I think in this data set it takes about, I don't know, between 5 and 10 minutes to run with only 2 CPUs. Well, I'll start it just to show. it will tell you that it went into the directory, so that it found 41 images to analyze, and then you get this progress bar that shows you % complete. And that's even if it's running in like a remote cluster. And so the progress bar will start to fill up as soon as it starts doing the batch processing. It takes like a few seconds to sort of get ready, and then it should get started.
Noah, during this time, can I make a question?
Yep.
Are you guys planning make some click point interface for example, for fields documents? For example, just I don't know, any Java interface, something that the user just instead see the black window that scary many people. You just say there, oh, change this document, and then the person just type the name, and at the end the output is exactly this morphology workflow configuration JSON.
Whoa. Sorry, I stopped that. I think in this environment it's printing out weird. Good question. So one thing I would definitely like, so Haley showed the custom ROI, right? You had to define these XY coordinates for the vertices of the polygon that she drew around the plant. I think we could definitely, like you do in field image R, I think we could make an interface in Jupyter where you could just click on the image at points and not have to write down the coordinates, where it could collect those automatically. I think that would be straightforward. We do get asked a lot about like a graphical user interface for PlantCV, and it's a good question. I mean, actually, if someone was really interested in doing that, we would totally support it. It's not our area of expertise, so we haven't. But if people have ideas too about how to maybe for this configuration part too, I could see what you're saying about sort of an easy form to fill out instead of editing a text file.
Do you remember the Shine workshop? Basically, it's programming in R, but using a Shine interface click point. I guess it's a really good idea too. Okay, sure. Thank you.
So then let's pretend that ran for 5 minutes or so. We would get this output file. This is what we defined. So sorghum results.json. It's a really ugly file to look at. I'll click on it. It's an endless single string of all this text. So it's not meant to be readable, but it stores the data in a kind of useful way, but not good for analysis. So the last part that we want to do is convert that into output files we can actually use in something like R or Python or whatever you do your analysis, even Excel. So another program that comes in PlantCV is called PlantCV-utils.py, and it has a subprogram called JSON to CSV. I have to remind myself what the inputs are. Right. So it has two -j JSON files. So that would be like sorghum results.json. And then -c is the prefix to the output. I'm going to say test for now because I don't want to overwrite the files we already created. But if I run that, it just takes a second, and back over here. It created these two files, so test single value traits.csv, test multi-value traits.csv. But the real ones that were created before that are available already are here. So if I click on the single value one, it's still kind of a wide table, so it's hard to see here. But it has columns for all of the metadata we collected. And then it has Oh, this one. Okay, sorry. I think we had other measurements in a different iteration of this, but however many analyze functions you ran, it's totally up to you as the user. So all the things that you measured would be stored in columns. Here, in this example, it's just stem height and stem angle and stem length. And then in the multi-value one, we would have, this is already formatted for something like ggplot or other kind of tidyverse tools. So I think we might need to fix this. I think this is an incomplete file. All right. Sorry, let me go to this one. Oh, no. Sorry. My bad. This one, the last couple columns are trait, value, and label. So the traits themselves are defined like down in the rows. So obviously it's hard to read here with like my text being as big as it is, but well, yeah, anyhow, it's hard to read, but all the things you would want are down there. So then you could filter that table, like if you were only interested in like the leaf angles, you could extract those out of there in R. So as a quick example, Haley created one more notebook just to keep it in Python, where she's reading in the multi-value traits output, and then we're just doing a couple quick plots. So we won't go through all this, but we're just going to use pandas. We're going to do something like R, but just with Python instead. And we just want to look at a couple of the quick things. So we can just measure, like as an example here, the mean leaf angle in degrees for the two genotypes that this subset of data had. So you can see that one of them is almost 50-degree insertion angle, so the angle between the stem and where the leaf goes into the stem. And then the other one's more like 45 degrees. Now that's the average across all leaves, so right. Right, and then for any other trait, you could measure like leaf length or anything you could do data analysis on. I will also mention that inherently, like these data are often time series, and so all of the time measurements for all the time points are also in the output data set. And for the morphology stuff, the trick is, and this is something we're working on now, is you get measurements for each leaf at each time point, but in order to make them even more useful, we want to be able to assign the identity of the leaves between time points. So that's something we're actively working on. We're finishing up what we call PlantCV version 3 series, and in PlantCV version 4, one of the new sub-packages will be dedicated to time series analysis, where we take an entire time series of images for a plant and get even more information out by not just considering them individual images at individual times, but using that time series in a more interesting way. So I know this last part was quick, so if you end up having any questions about that, definitely just contact us. all of the pre-computed results are in the GitHub repository. So so they're there, and the shell commands that I ran are in this file called shellcommands.sh. So the PlantCV workflow and the PlantCV utils JSON to CSV commands are in there if you want to try running that on your own. So definitely appreciate everybody who was able to attend or watch later. And thanks, Felipe and Anarita, for having us.
Yeah, thank you guys for giving this very interesting workshop. We had many comments, and we are very happy of that.
I'm gonna invite the other speakers to be here, and then we have the chat. Yeah, great. Yeah, guys, thank you. You're doing a really great job. And thinking about this new version, you said that there is new tools, right? And what, for example, these new tools can be used now? And for example,
really good question. So we openly develop on GitHub, meaning that despite the numbered versions, like the newest version is always on GitHub. So if you want to live on the bleeding edge, you can get PlantCV from the source code, and you can be part of the discussion about that in the GitHub issues, or on however you want to contact us. Just during the transition between 3 and 4, some of the new stuff like the time series subpackage exists as a pull request right now. So meaning it's sort of in development in a separate from the default branch. That doesn't mean you couldn't use it though. I mean, it's up on GitHub. Just be aware that it's, I don't know if you call it beta, alpha.
Yeah, I want just to say that based on my experience, if you guys are, so I was not a Python programmer before, so I think don't get scared, because PlantCV is very easy to learn, and this guy, they helped me a lot. So if you are trying to develop your workflow and you have errors, you can just post them on the GitHub issue, and they really help you to solve them. And so yeah, that's based on my experience. And there is a question if you guys want to answer. I will say the last one. I'm curious how it's possible to integrate PlantCV with other tools in one pipeline.
Right. So because we build on the Python scientific communities tools, like Malia mentioned, the images are NumPy arrays, and almost all of the image analysis tools in the Python community utilize that NumPy data structure as the core element. And so if you're comfortable with it, you can definitely use OpenCV, scikit-image, scikit-learn directly. And because the workflow is just a Python script, you can combine any other tools you want into your workflow. That requires obviously like going and learning about all those other packages. But that is there, and you can make your workflows as complicated as you want to. And if you discover things that you think would be useful in PlantCV, then that's a great way to sort of test it out for yourself, and then build it into something like a function that other people could use then.
Yeah. Well, thank you guys. I think we are already over the time, but we had a lot of we really enjoyed this workshop, and I want just to say that they are on our Slack workplace. So if you want to ask any question, you can there, but also I'm sure they will be happy to answer also if you contact them directly through the GitHub issue page or their email, institutional email. Yeah, thank you guys. And
yeah, I just want to say thank you too. And if you want to use this time to talk with students and invite people to be part of your research, so use this time now to say thank you for the group of people that's following you guys. There is a huge group. Thank you.
Thank you both. Yeah.
Thanks, guys.
Thanks so much for hosting us.
yeah. Okay, so I think we are ready to move to move
Final part, right?
To the last part of our workshop. So this is like the list. Oh, we also want to say that just this week we got another workshop schedule for the was the 20?
Yes, it's gonna be November 20th. It's our last workshop, and it's gonna be with Larry York from Nobel Research Institute.
Yeah, he's gonna introduce his software for analyzing roots. We understood that this will be a great tool for many people, and we also understood that there are not so many talks about root, and so we wanted to include them. And so we will add him in the list and also like in the registration link. So if you are interested, you can register to that as well. And now is the time to introduce our next speakers. For next Friday, he is Eric Chang, a graduate research assistant at Ohio State University. So he's gonna introduce us to Tide Models, which is a package for machine learning. And he's gonna show us how to use this package for plant phenotyping. So same times, same registration link. If you already registered for this workshop, you will not need to register again because we send the information every Thursday as we have done so far. But of course, you are welcome to invite anyone that you think can be interested. Yeah, so thanks.
Yeah, guys, thank you for being here and being part of this project with us. Send your comments, send your suggestions. We are here to learn and share with each other. So
yeah,
thank you and see you next Friday. Bye-bye.
Bye. Bye-bye.